Bom dia a todos, obrigado por participarem desse nosso webinar conjunto da Câmara Brasil-Israel, o Instituto Weizmann e o Technion Brasil, cujo tema é muito interessante, muito atual, que é a Medicina do Futuro. E hoje temos o prazer de receber dois professores renomados, Shulamit Levenberg, do Technion de Israel, e Ronny Paz, do Weizmann Instituto de Israel. Shulamit, Rony, good morning here. Thank you very much for joining our webinar. It's a pleasure to have both of you here with us. Eu queria fazer um agradecimento também especial a todos que colaboraram, que conseguiram fazer esse evento, em que pese toda a situação de Israel, que está muito difícil nessas últimas semanas. Eu queria agradecer em nome da pessoa da Cila Schumann, que coordenou tudo isso também. Eu queria agradecer a todos, diretores, colaboradores da Câmara, do Weizmann e do Technion, para que tudo isso acontecesse. Muito obrigado. A Câmara, juntamente com o Weizmann e o Technion, a Câmara é uma entidade cujo objetivo é criar, desenvolver e aprimorar negócios e oportunidades entre empresas privadas brasileiras, investidores privados brasileiros, com empresas privadas de Israel e fundos de VC para investimentos. Tem desenvolvido aqui no Brasil um trabalho grande, um esforço muito grande para aprimorar cada vez mais esses negócios, e principalmente na área de tecnologia, na área de medicina e de ciência. Por isso nós estamos fazendo mais esse webinar conjunto. Queria agradecer o Weizmann na pessoa do Mário Fleck, que é nosso presidente do Conselho da Câmara Brasil-Israel, que nos honra, é membro do Executive Committee do Weizmann Institute em Israel, e ao Salomão Iosp, nosso amigo, presidente do Technion Instituto no Brasil. E queria algumas palavras do Mário e do Salomão. Mário, por favor, fique à vontade.
Bom dia a todos. Renato, obrigado pela oportunidade. Uma alegria muito grande participar desse evento com três instituições que eu admiro tanto. A Câmara Brasil-Israel, pelo seu papel estratégico fundamental nessa relação bilateral muito importante, e, evidentemente, duas instituições que são orgulho para todos nós, tanto o Technion quanto o Instituto Weizmann. Particularmente, algumas palavras sobre o Weizmann, que eu conheço há bastante tempo. O Weizmann é um instituto de ciências básica, né? Ciência pura, portanto, o seu primeiro estágio de desenvolvimento foi uma instituição criada pelo Ronny Weizmann, que todos conhecem, foi um cientista, diplomata e primeiro presidente do Estado de Israel, que teve um papel fundamental na sua criação e teve uma visão muito importante, de longuíssimo prazo, a ponto de criar, anos ainda antes da formação, da criação da independência do Estado de Israel, da criação de um Instituto de Ciências, uma universidade do porte do Weizmann, assim como ele também fez com a Universidade Hebraica de Jerusalém. O Weizmann tem algumas características especiais interessantes. Primeiro, que é uma preocupação focada muito maior no cientista do que na ciência, pensando que a ciência vai ser sempre uma consequência de cientistas de excelência, movidos à sua liberdade para explorar toda a sua curiosidade e desenvolver com materiais e com laboratórios de qualidade e excelência o maior avanço possível na pesquisa científica. Dentro dos campos tradicionais de física, química, biologia, mas também matemática e ciências computacionais, num ambiente muito interligado, multidisciplinar. Então, a ciência no Weizmann permite uma conexão entre todos os seus departamentos com muita facilidade e com muita intensidade. E hoje, como consequência desse ambiente, a gente tem, por exemplo, dos 20 remédios mais vendidos, uma pesquisa recentemente mostrou, mais vendidos no mundo, 7 foram patentes desenvolvidas a partir da ciência básica feita no Weizmann. Então, com muito orgulho aqui que eu represento o Weizmann aqui no Brasil, tenho uma participação intensa no Executive Board, e tenho muita alegria de trazer para cá também, para esse evento, o professor Ronny Paz, que certamente vai fazer uma palestra muito interessante para todos nós. Obrigado, Renato. Obrigado à Câmara.
Obrigado, Mário. Muito obrigado. Salomão Iospe, por favor, crie umas palavras suas sobre o nosso webinar e um pouquinho sobre o Technion no Brasil, que é uma coisa nova, né? Recém-criada por você, ideia brilhante sua, e eu acho que é bem interessante se explicar um pouquinho para todos.
Bom dia, Renato. Bom dia, Mário. Bom dia a todos. Bom dia a todas. Eu sou Salomão Iospe, eu sou presentemente o presidente da Associação de Amigos do Technion Brasil. Essa associação também é conhecida como Brazilian Technion Society. A Associação de Amigos do Technion Brasil, como o nome diz, nós somos um grupo de amigos do Technion que visa dar suporte às atividades do Technion, trazendo o Technion para dentro do Brasil, criando espaços de colaboração entre o Technion e entidades da sociedade brasileira, tanto universidades públicas, privadas, centros de pesquisa e também empresas. Eu estou muito feliz de estar aqui hoje participando desse evento tão primoroso da BRIL Chamber, capitaneada aqui pelo Renato, que nós estamos trazendo a professora Shulamit Levenberg, que foi a diretora por muitos anos da Escola de Engenharia Biomédica do Technion Israel Institute. Deixa eu falar um pouquinho, então, do Technion. O Technion, ele foi a primeira universidade de Israel, foi fundada em 1912, muito antes, então, da criação do Estado de Israel. Um dos fundadores é ninguém menos do que Albert Einstein. Já na primeira turma de engenheiros formado pelo Technion, você já tinha uma mulher, isso no início da década de 20. Então, vocês veem como é uma entidade visionária. A maior parte dos engenheiros de Israel saiu do Technion, e a missão do Technion foi ter as bases da infraestrutura desse Estado que seria criado em 1948. E assim foi. Não estamos falando só de pontes e construções, estradas e redes elétricas, estamos falando também de água, por exemplo, que é um bem escasso e tão importante para o Estado de Israel. À medida em que o Estado de Israel foi fundado, o primeiro desafio foi a sobrevivência desse Estado, e o Technion foi fundamental, por exemplo, em engenharia aeronáutica, na criação das potencialidades e das competências, por exemplo, na questão aeronáutica e aeroespacial. O Technion também, nos anos 60, além das engenharias e ciência básica, começou a ter a medicina dentro do Technion, e você teve uma visão do Technion de que dentro da medicina a inovação tecnológica seria fundamental, e daí que foi criada a engenharia biomédica. Mais tarde, o Technion se preocupou muito com a transição entre ciência, inovação e empresas e produtos, algo que hoje a gente chama de startups. E Israel é conhecido como Startup Nation. Não haveria Startup Nation se não tivesse havido o Technion. O Technion foi o embrião dessa Startup Nation em muitos sentidos. Não é por nada menos que você tem quase 50% das empresas de Israel tradeadas no Nasdaq, que é a bolsa em Nova York, onde são tradeadas as ações das empresas de tecnologia. 50% aproximadamente são de ex-alunos ou professores do Technion ou são lideradas por esses. Outro dado importante sobre o Technion é que o Technion, a partir dos anos 2000, começou a se internacionalizar. Hoje, o Technion, além do seu campus em Haifa, tem um campus em Nova York, juntamente com a Universidade de Cornell, no centro de Nova York, bem como um campus na província de Guangdong, China. Então, o Technion passou a ter uma presença internacional muito grande. Então, em resumo, o Technion, ele, além de abrigar as ciências básicas, como física, química, biologia, matemática e também ciência da computação, abriga as engenharias, civil, mecânica, elétrica, engenharia de alimentos, indo até engenharia biomédica e outras, arquitetura também, e medicina. E hoje em dia existe um termo que está sendo usado, vamos chamar isso de um buzzword que a gente tem ouvido, de consultorias como McKinsey, Boston Consulting Group, que se chama, o pessoal fala de Deep Tech. Deep Tech é você conseguir quebrar aquele ciclo normal de ciência básica. Depois de ciência básica, você começa a ter aplicações que utilizam ciência básica, e depois você vai ter design, que vai criar, então, um novo produto. Deep Tech é nada mais do que você acelerar isso exponencialmente e fazer tudo isso ao mesmo tempo. Então, o Technion, contando, inclusive, com 3 laureados do Prêmio Nobel em Ciências, ele é um exemplo disso. Está no DNA do Technion, a criação de novas empresas, novos produtos, enfim, de inovação tecnológica. Então, obrigado, Renato, obrigado, Mário, prazer estar aqui com vocês. Passo a palavra para você, Renato.
Obrigado, Salomão, obrigado, Mário, pela apresentação ilustrativa sobre as duas entidades, muito importante, e obrigado aí por todo o esforço, toda a dedicação que vocês dois fazem por essas instituições aqui no Brasil. Let's move to English, and we have now our guest speakers. First, Shulamit Levenberg. Shulamit is going to say about tissue engineering and 3D printing for regenerative medicine. Shulamit, she's a former dean of the Faculty of Biomedical Engineering of Technion Institute. I was reading your curriculum. It's so extensive. We stayed the whole webinar reading your curriculum. Your and Ronny also is okay. So, but anyway, Shulamit has many prizes, awards all over the world. And very important, she's founder of 3 startups of companies in the areas of culture meat, spinal cord, and regeneration. Shulamit, thank you again for joining our webinar, and please, you have the floor from now on.
Okay. Thank you very much, and I would like to present some of our work on tissue engineering. And if I can have the slides presented. Thank you very much. So I will focus on how tissue engineering can help us both in biomedicine, and also I will mention how it can also be relevant for food technology. So if we start on with why we need tissue engineering and how tissue engineering started. So the need is for tissues and organs for implantation, because we know that if there is an injury and an organ is not functioning, or any disease, or tissue that is broken, a bone or spinal cord, so the options are either, you have nothing to do, or sometimes you can get a tissue or organ from donors and implant it, but then you have to make sure you have donations, but also the recipient will have to get immunosuppression through all his life, which is really very problematic. And another option is to take from the patient, from the person, but to take tissue from one place to repair another area, the damaged area. You can take bone from one leg to repair damaged bone, you can take muscle from one place to the other, or you can take a vein for bypass operation. But then, of course, you have, instead of one injury, you have two sites that are now injured, and the surgeries are long and complicated. And in tissue engineering, the hope is to be able to take cells from the patients and then expand them in the labs. So you need, many cells to build a tissue, or especially an organ. But then after expanding the cells, you can, and if you start with stem cells, you can really get high number of cells. Then you can grow them on 3D matrices, on 3D scaffold, we call them. These can be sponges-like or gels, and the cells will be seeded into it. They will start to organize and differentiate and mature into the tissue specific organization, and then you can have the tissue and later on the organ that can be implanted to the same patient. And since we know that each tissue is actually composed of by several cell types, not only one, we can also do it. One second. We can also do it also in the lab. We can now mix several cell types together in the lab. And for example, if we're talking about muscle tissue, we can take muscle cells, we can take endothelial cells, we can take support cells, and now mix these cells together in the tube, and before we seed them onto the scaffold. And of course, we have to find the right cell ratio and the right medium composition that will fit all the cells. But now we can mix the cells together and seed them onto, here you can see a sponge-like scaffold where the cells are getting into the pores of the scaffold, and the cells will start to organize into the tissue. But we all know that if we want to implant the tissue, it needs to get oxygen and nutrient after implantation. And we and others actually found that if we add endothelial cells, they will form spontaneously tube-like structure and will resemble the blood vessel network that we see in a tissue. So you can see here how the tubes are being formed inside the scaffold. And if you add support cells, some of them will differentiate to pericyte-like cells or smooth muscle-like cells. They will be recruited to the vessel wall and will stabilize the vessel. And if we add myoblasts, they can differentiate to myotubes, and we will have the muscle fibers being formed in the tissue. So we have muscle tissue, and within it, we can have the vascular network, which is very important. And if we have you can see it here, right, 1 second. If we have the tissue that now we want to implant it, how we can really help. For example, here you can see a damaged muscle, the abdominal wall muscle piece is taken out, and instead of it, we can suture the engineered tissue. It can be vascularized, it can be with all the vessels that we formed in the lab. And now this vasculature will induce penetration of the host vessels into the implant because they provide signaling, but also because we will see nice connection, nice anastomosis between the two types of vessels, and some of the blood now will be flown into the inner part of the construct because the engineered vessels will become functional, and this will also help to provide faster oxygen and nutrient into the implant. So this is how it looks under the microscope. You can see the implant area, the cells, the engineered vessels are in green. Everything here is filled with other cells, but we only see in the implant the engineered vessels because these were labeled. And we can also label the host vessel in blue. And we can also inject a fluorescent dye, red fluorescent dye, that will show us which vessels are really being perfused, are really being functioned. And in a larger view, you can see very nicely, 1 second. You can see very nicely the connection between the engineered vessels in green and the host vasculature in blue. And now we have to look to see if we have the red dye going all the way through the blue to the green. And indeed, you can see that some of the engineered vessels have the red dye inside them, meaning that they are connected and becoming functional and helping to perfuse the blood faster into the inner part of the implant. And this is really very critical in tissue engineering. In another study, we were able to show that we can now also vascularize bone tissue, so we can have composite tissue of soft tissue and bone tissue. And I think you can see here very nicely how the vessels sprout and really vascularizing the bone, and we can now use this bone to repair a damaged bone. So, and now if we move to, if we want to make the tissue more complex, more complicated, more, with more, we can print the cells and the scaffold together and define exactly the structure of the tissue that we want, and we can really have it fit the exact size and structure of the injury site. So what you see here are the bioprinter, where you print bioink. It's not a regular printer. What you print are either the scaffold, and you can also print the cells together. And we established a bioprinting center at the Technion. This is one printer. We have other printers showing, for example, here that you can make larger tubes, and you can also have the self-assembled vessels, as you saw in the movie before, that the cells self-assemble and form the network, and we can have the tissue built with different cell type now. With a printer, you can go into larger pieces. Here you can see another printer that we have in the lab, and with robotic arm, and we can have more structure of the vessels. You can see here, instead of one tube, we can have the whole array of the tree of the vasculature, and we can also add more printing method, like printing inside a bath, that hold the tissue together as the bioink is really being solidified. So you print the bioink, and it really get polymerized by different techniques, and eventually you can get the scaffold being built and the cell being printed at the same time. As this is really bring the tissue engineering toward the clinic by being able to print the tissue which will be more similar to the tissue of interest. So what we can do with tissues like this, that we vascularize and make them very similar and make them also functioning. So for example, one area is to treat diabetes. We were able to show that if we vascularize pancreatic islets, we can now implant this scaffold, the vascularized islet, we can implant it under the skin, and the cells will now secrete insulin because they really have all the blood supply. They will function better. They will secrete insulin in response to glucose, and we can see that we can repair diabetic situation in the mice. But we can also do it in another way. We can also now use tissue engineering, muscle tissue engineering, as a novel way to cure diabetes. And you can see a project that we did with a thank of donation from the Schneur Foundation. We have an idea, and we're able now to prove it with this donation. So we took the engineered muscle, and we engineered the cells to express the transporter, the glucose transporter, and we're able to show that if we take this implant and implant it in the muscle, it can express the transporter and now absorb glucose in a better way. And you can see here the implant expressing the transporter and showing here that indeed the blood glucose level of the diabetic mice really was reduced, meaning that these small pieces of muscle tissue, engineered tissue with engineered cells inside it, can now function to reduce the blood glucose levels. And this can have also systemic effect and also affect other tissues. For example, you can see here the effect on the liver, that now we have less lipids in the liver, meaning that there is a systemic effect to this small piece of engineered muscle tissue that is now functioning to repair disease, actually. This is another model of model showing diabetes. Now you can see the insulin affecting the insulin sensitivity, similar to what you have in diabetic patient. And also here, we can see that we can stabilize the glucose level by such an engineered muscle implant. Another area where this can be very important is this tissue engineering technique that we're working on it in the lab. Another area is spinal cord regeneration. And we were approached both by the Israel Spinal Cord Injury Foundation, and also we got a donation from Australia and from Shervington Fund to study how we can use tissue engineering to repair a damaged spinal cord. And what we did, we decided to use a engineered construct with stem cells that we can implant in the injury site. And you can see here in the movie that the rats will, I don't know why it's not moving here. One second, let's see. The rats are paralyzed and they cannot walk without the regeneration, without the implantation of the engineered construct. And what we did, we saw that after implantation, so I'm sorry, this movie, we'll move to the next one. You can see here that after implantation, the rats that really were paralyzed in the back leg started to walk again. They stand on their leg and started to walk only by the implantation of the engineered tissue that provided neurotrophic signals and allowed the regeneration of the axon. So this was really very exciting to see that we can use tissue engineering to induce regeneration and induce repair, to the extent that we really hope that we'll be able to move this into patients. In the spinal cord injury, we also use vascularization, similar to what you saw before. We can add endothelial cells, they can form the vessels, and this was also shown to help in repair. Especially we could see more sensory recovery by this vascularization, many vessels that are now induced to form in the injury site because we added endothelial cells to the construct. And another area which we now explore are exosomes that we can deliver by nasal administration, intranasal administration, and the exosomes are modified with siRNA that provide actually signals for axon to regenerate. And by this, we could also see very nice repair. You can see it here, the green line showing the repair of the rats. And by this, we also now founded a company because this is less invasive than in implantation of the engineered construct. We can now get the exosomes and give them through the nose, and they reach the damaged area and also bring the rats back to walking again. So we can see that tissue engineering can help in regeneration. We can see different models of bone and spinal cord, pancreas repair. But tissue engineering can also be important in other directions. And I can show here the 3 companies that we founded. One is Aleph Farm, that actually used this technique of tissue engineering, of skeletal muscle tissue engineering, to engineer cultured meat. I mentioned Norexon, the company for using modified exosome for spinal cord regeneration, and also NanoSynex, that we develop a microfluidic device and we're using it for antibiotic susceptibility tests. And actually one of the founders, she is from Brazil, and we're very proud of the 2 young entrepreneurs that came and established this company from the technology that was developed in the lab. So regarding how we can use tissue engineering to cultivate meat. So the same concept, we can take cells from the cow and we can culture the cells, grow them on scaffold. Now the scaffold will be edible scaffold, and we can grow a piece of meat, and we'll be able to show that we really can do it. We can also mix several cell types together, and we can form very nice fibers, muscle fibers in the dish. And this can now be, we move to making it more similar to the tissue, to the muscle tissue, by adding different cell types together, the endothelial cells and support cells and the muscle. All of it enhance the muscle differentiation. We can get a nice piece of differentiated muscle. And based off this technology, we founded Aleph Farm, that actually starting to prepare this as a piece of, these are sim-cut steaks. And also using the bioprinting, we moved to also use it for printing larger and thicker steak cuts. You can see here, these are the thin cuts. and chef was, plated it, and people tasted it and say that it's very similar to the regular meat, although it was made outside the cow without harming the cow. And using the 3D printing, we now used and made this piece of steak with fat areas and muscle area, mimicking ribeye steak by adding to the tissue engineering also the technology of bioprinting with a special bio ink and using fat and muscle cells to prepare the steak that was also cooked and very similar to a whole cut steak. So I will end here. Of course, I would like to thank my group, all the students that were involved, all the funding, and also all our collaborators, around the world and also here in Israel. And I would like to thank you also for listening.
Thank you, Shulamit. Very interesting presentation. And now we have Professor Ronny Paz. Ronny, thank you very much again for joining our webinar. Sure. Ronny is going to talk about the emotional brain and traumatic memories. Ronny also have an extensive CV, and he's head of the Department of Neurobiology at Weizmann Institute of Science. He got many awards, and he studies the interactions between emotions and cognitive process, such as learning, memory, and decision-making. So, and just an information for the audience that we have the chat, and the question can be made in the chat, and we will ask to the guests, speakers after Ronny. Okay, please, it's your time. Thank you very much.
Thank you. Do you see my presentation?
Yes.
Good. Okay, thanks. Thanks a lot for inviting me. It's going to be very hard after Shulamit gave such a beautiful and highly interesting and innovative talk. So I'll do my best. So thanks a lot for inviting me. So I'll start by actually giving you some background about what we're doing now at Weizmann, and then I'll dive a little bit into my own research. And so we're actually now in the progress of establishing a new neuroscience institute, and this new institute is going to be a new building, and everything is going to be all targeted at understanding brain and neural sciences. There are going to be about 40 labs in this new building that all are going to do research about brain sciences. And the main reason is that, as most of you know, both psychiatry, mental health problems, but also neurodegeneration. Shulamit touched a little bit about some of the problems and how and her ideas to solve this. And so what we know is that if we look at, I'll talk mainly about mental health, if we look at depression, anxiety, post-trauma, but also Parkinson and dementia, and similarly, there's an increase. Although we advance in medicine and research, there is an increase all across the world in the number of cases of depression, anxiety, autism, post-trauma, etc., etc. And so this is puzzling. How come there's constantly increase in the cases? And we can even say that COVID-19, the last year, has made it even worse because there are a lot of side effects or main effects of COVID-19. There are long-term effects of the illness. Many people lost their jobs. There's a lot of stress, definitely social solitude. This all will lead soon to what we call the second pandemic, which we are certain there's already research about this. There are going to be many more mental health problems such as depression, anxiety, and trauma. And what I'm going to say today, I think, and I think you'll all agree with me, is that the only way forward is basic research. There's been no real change in treatment for these diseases in the past 30, even 40 years. It's just the same kind of drug medicine that has been found by accident like 30 or 40 years ago, and there's no real change. And I think one thing we can learn from COVID-19 is that we should invest a lot in basic research, and that's a good lesson, because if we talk about COVID-19, then the vaccination came really from basic research of mRNA, which I'm sure you heard of, you all heard about. So there have been like 30-40 years that we know about mRNA, and it's taken a lot of time and research, and it was driven initially only by curiosity into cell biology. And the result, which we saw in the past year, was that we had a record time in developing the vaccine. I think that's an important lesson for all of us, for researchers, but also for everyone, that we need to invest in basic research, because it's going to help health problems when they come in 5 years, in 20 years, or whenever. So I think that's a good lesson. And so our role is trying to understand how the brain generates what we call adaptive flexible behavior, and that is what makes us human on one hand, and what breaks us, what leads to disease on the other hand. And for this, we're now setting up the new flagship project at the Institute, at Weizmann Institute, the Institute for Brain and Neural Sciences. We already have a building planned with, as I said, 40 labs. And I'm highlighting there are 11 centers of research. I'm highlighting the Neurodegeneration Center because this is actually the Brazil Center for Neurodegeneration. It's a beautiful and very kind donation, and thank you all for that. I'm sure you can get more details about this if you want. And what I'm going to talk today is about a different center and my topic, which is mental and emotional health. And so I'm going to dive into science now, and the topic of my talk is the emotional brain and traumatic memories. And mainly the question that my lab is interested in is, is why evolutionary adaptive emotional memories actually become a trauma, post-traumatic stress disorders. Because we can think about emotional memories as a good thing, right? If you had a very important emotional bad memory, you want to remember this forever because you want to have better survival. So if there was any kind of an explosion or an animal, a predator, you want to remember all those things forever because you don't want to lose your life the next time. And so these are basically what we call rational adaptive behaviors. But in extreme cases, and for reasons which we are now starting to understand, sometimes these rational emotional adaptive behaviors become traumatic and cause anxiety, post-trauma, and depression. And I'm going to discuss today three models that we developed in the lab by which we classify the PTSD and depression into different factors, behavioral factors, but also brain mechanism that lead to what we call the maladaptive response, the traumatic response, or the depression or anxiety. So one such mechanism is a mechanism of uncertainty and surprise. And I will not go through all the details, but you can see the 9/11 famous events on the right. And for some reasons, although for some people, approximately 5 to 7% of the people who actually saw or heard closely about the 9/11 bombings, Twin Towers, for some of them, although the memory of the building, of the explosions and everything do not go away, although they still go around in the world and see many other safe, tall buildings. And so we call that in the terminology extinction. Why did these people do not extinguish the traumatic memory? And so we developed a model in the lab for this, and we could show that if we have learning that is driven by some kind of surprise or uncertainty, which was just the case in the 9/11 and also for many other traumatic events, they come as a surprise to the person or to the animal. And so when a memory, when there is learning that is driven by high surprise or uncertainty, because you cannot predict that this is going to come soon, then there are two regions that play an important role, and you can see the two regions in the middle. One of them is the amygdala, which is a small region in our brain. Basically, if you go from the eye inside and from the ear inside, it's where the two lines meet. And there's the prefrontal cortex, and there's a very gentle balance between those two regions, where the amygdala basically signals, neurons in the amygdala signal that there's a bad traumatic memory, and the prefrontal cortex should learn safety, meaning should learn over time that it's no longer a traumatic memory. And for some reasons, for reasons which we were able to identify in these papers, when a memory is driven by uncertainty and surprise, the prefrontal cortex can no longer tell the amygdala, deliver neural signals and tell the amygdala that it's already a safe memory, and therefore we still have a traumatic memory, and that is unmodulated. And so we did that by developing new technology of recording simultaneously in the two brain regions in 2 to 3 different species, and show that short-term correlations between teaching signals in those two regions is what led to the memory that resists extinction and therefore can remain a traumatic memory if it was acquired under a surprise. And another important model that also underlies a different group of post-trauma and depression patients is what we call memories that overgeneralize. And so you can see on the upper right, again, you have the, for example, this is obviously an example, but we found this in a real population of PTSD patients. If you experienced 9/11 or any other memory, then some people, we can call them normal, whatever that means, a normal response, is that in the case of very similar environments, in this case, high buildings is an example, they will show, express distress and trauma and anxiety, and that makes sense because it's similar enough to the original environment when the memory was acquired. But in post-trauma and anxiety patients, you'll have, as the red there, what we call wider generalization. And so even in buildings that are much smaller and or do not look the same at all, they will still experience this kind and show trauma and stress and anxiety. This is obviously a simplification here with the height of the buildings, but it can be shown that this overgeneralization happens over many high-dimensional factors of the stimulus, and environments look similar. And so we found that this is due to real plasticity in the brain. I'm not going into details now, but we were able to show that this is due to actual plasticity in networks in the human brain that actually result in perceptual discrimination, changes in the discrimination threshold. So if you previously considered only this kind of building to be as the previous one, you will now think that even smaller buildings look like the 9/11 in terms of height. And the important thing is that this is subconscious. It's not something that you can explicitly say. It's actual perceptual plasticity. So you do not your brain cannot differentiate between the two signals. And we repeated this and tested this in populations of anxiety and post-trauma patients. And what we could show is that there's indeed a generalization, a wider generalization around something that was aversively conditioned, kind of like a traumatic memory. And this was directly linked and correlated with activity in the amygdala, which you can see in red on the other side. This is using imaging in functional imaging in patients and in humans. And so we could show that the amygdala underlies, just as before, amygdala here underlies the wider generalization. So it's the same brain circuit, but different mechanism. And the third mechanism that we are now working on very extensively has to do with what we call the exploration-exploitation dilemma. I'll explain this in kind of broader terminology. And so we all animals are facing this exploration-exploitation dilemma. And the best way to explain this is to show this by some kind of a restaurant. And so if you know you have a good restaurant outside, you can always go to it and have a safe food. But if there's a new restaurant, you also want to decide whether you kind of take the chance and go to explore this new restaurant because it might be better. But you also take a risk because it might be worse and you're better off going to the old good one. And so this is obviously kind of a simple, straightforward example. But if you think about it, then it comes for a very rich survival, animal survival and evolutionary record. Animals have to always exploit their environment. And we also do that for any kind of decision making. Do we want to quit our current job and look for a better job? Well, on one hand, this job is not perfect, so maybe look for a better job, but it's obviously a risk. And looking for another job because you lose this one. You can always say that looking for a partner for life is a similar decision. You always have to make some balance. And so we thought that this very simple decision-making task can actually underlie a lot of the anxiety and post-trauma effects. And so we did this and tested this. And what we could show very nicely is that exploration, the rate of how much you decide to explore, goes high the more the anxious individual is, and that actually harms decision making because the best, you can see that on the well, there's an inverse U-shaped curve. The best decision making strategy comes when you do medium exploration, and that comes when you do when you have medium anxiety. And not too much too high, but also not too low. But we obviously care more about too high. And using, I won't go into the details, but using some statistical modeling, some machine learning modeling, reinforcement learning based, to be more precise, we could show that there are two different systems in the brain. One of them care about value, and so actually anxious individual care less about the value, which happens in the dorsal anterior cingulate cortex, and they care more about uncertainty. So it's not so much about how much I think I'm going to get from the next option. It's that I don't know what happens in the other options, and that drives me to explore more. So these are three behavioral models: resistance to extinction, overgeneralization, and overexploration, that we could show that are all underlie increase the chance of developing a post-trauma anxiety or depression. And so our next stage was kind of to think, can we actually manipulate the emotional memories and kind of help patients? And our goal was to do it using deep brain stimulation with DBS, which is, some of you might have heard, it's something that, for example, in Parkinson happens a lot. Well, not a lot, but people do these surgeries in Parkinson. It's now being tested for depression as well, and we're developing this kind of models for depression to do this. And so we first tested it in rodents with new tools, with optogenetic tools, where you can specifically manipulate a specific neural pathway. That's the benefit of optogenetics. You can inject in the amygdala and then shed light on the prefrontal cortex. And so we know we specifically manipulate neural signals from the prefrontal cortex to the amygdala. and we could show that it actually enhances the extinction of traumatic memory. But we wanted to see, obviously, this is in rodents and very nice. We wanted to see if we can do that in primates as well. And so this is kind of, I would say, the goal. And so this was done in non-human primates. We actually put an electrode in the cingulate cortex, and we stimulate during extinction learning. You can see it on below. And what we could show is that if we use a specific type of low-frequency stimulation, which actually reduces excitability in the prefrontal cortex, then the traumatic memory does not return after 24 or 72 hours, and the animal had a good extinction protocol, and the memory did not come back. And we're now actually trying this in collaborations with neurosurgeon in Israel, 2 centers, to do this in patients that have electrodes implanted. I will mention two other things that we work on. And so one thing, sorry, let me go back. Wait, let's go back because I want to show this again. Okay, yeah. So the next stage is obviously, so everything I showed you was done on what we call classical conditioning models. But obviously, as humans, we can learn many more sophisticated things. It's actually a hallmark of the evolution. And so together with Elad Schneidman, a professor here, a physicist and mathematician, we developed a new framework of how we can look at learning different rules, which are obviously the most sophisticated things we can learn. And we could show that using a very sophisticated geometrical representation, we can describe learning of new rules and identify how they react in 2 different regions of the brain, and we could see the different roles. And we can actually now do a specific fine-tuning for brain enhancement, for learning that is brain enhancement driven. We can measure the neural activity and know what to present next so that people can learn better. And it obviously has a lot of relevance for anxiety, PTSD, etc. We can improve the learning. We're still working on how we can actually stimulate in the brain to do this. This is going to be a little bit harder, but we think we're going to get there. And the final scientific thing I'll just say is that we actually have now a new 7 Tesla in the past year, 7 Tesla scanner, and we have, I think, the most innovative or advanced imaging facility in Israel, but in the Middle East, I would say. And many of the questions, this is done in collaboration with 2 new amazing neuroscientists, Michal and Rita, who are experts in how to induce plasticity using neuroimaging, real-time neurofeedback, we call it. So it's in the MRI, and we can actually use that to change the connectivity between different brain regions. And we're now testing how it can be used for autism and depression and all. And as I said, we're highly interested in the basic question of what are the limits of plasticity in the adult human brain. And I see it as 2 sides of the same coin, understanding basic fundamental questions and finding solving health problems. And so if you ask me what is my short-term or long-term vision, then we're now actually moving forward to test what we call emotional brain interfaces. And in this, that is very simple. We do a closed loop kind of iteration where the person, we do that both in animal models, but also starting now, as I said, in collaboration with neurosurgeons in humans, in human patients, where we do a specific behavioral stimuli model. We develop and record techniques and tools to read large populations of neurons. This is an effort all across the world, but do this here as well. And we use all kind of sophisticated algorithms to analyze what is really the neural code that underlies this specific behavior. And as a result, we can manipulate the specific brain patterns either via optogenetic or electrical stimulation as done traditionally to fix the network imbalance and hopefully improve the response to this behavioral stimulus. And so this is under constant construction. We actually have that working closed loop in non-human primates, and the next step is going to be in human patients. I think I'll stop here. I'll just say that the new Neuroscience Institute will have at least 35 to 40 groups within it. I'm showing here a few of them. On the left, you can see Alon Khan, who is our fearless president, and he's also a mental health depression expert, so that's good. And I think I'll stop here. I think, yeah, that's good. Thank you very much for listening.
Thank you very much, Ronny. It was a very interesting presentation, and I assume that I feel like I am 50 years ahead of our time. That's my feeling after the presentation of Shulamit and your presentation. And you are bringing many news for us. And we have now a very quick Q&A session, okay? We have a lot of questions coming in our chat on my private cell phone. And I will try to make a few questions for both of you, but we don't have enough time to make all the questions we have. And if you are available, we can make later on by email, and if you have time to answer the audience, okay? Shulamit, we have a question here. I will try to join two questions, okay? One is, are there ethical consideration or ethical restrictions regarding the printing of tissue or organs for replacing parts of the human body? And maybe you can answer with another one, is does the lack of movement of the meat made in the laboratory affect the texture? And if so, is there a way to fix it?
Okay, we start with the first question. I don't see any ethical concerns of preparing a tissue in the lab that can repair damaged tissue. It's just another way where we can save life or reduce pain and help in repair that we all try to do in the medical world. Regarding the other question, regarding the cultured meat, there is no need to have the muscle function. We want to have all the ingredients, the texture, and but not necessarily we need the functioning of the muscle for its taste.
Thank you. Ronny, question. What prompts mental illness? What promotes resilience? And how can we counter process that derail mental and emotional health? How can basic science contribute?
Oh, that's a very wide question, and it's a big question. What promotes? There are many factors that promote resilience. I'll say that as I did not mention the word, and the word itself, resilience. So thank you for raising it. This is one of the things that we and others are testing, and the question and the main. I think the main hypothesis now is that there are two factors. One is obviously genetics, and so, for example, this is the expertise of Alon Khan, who I mentioned, and the other is how the brain networks are wired during development, and that's something that I work on. And so I saw the question was also related to development, and that's a big part because, interestingly, the neural pathway between the amygdala and the prefrontal cortex keeps developing until the age of 21. It's the last synapse, it's the last neural pathway that actually stabilizes. And so everything that happens during development does not get modulated properly as it would have been if we experienced it as an adult. And so there's a big role for development. But the problem is that once it happened, the only thing we can do is go back into the network and try to understand how can we remodulate or rewire the connections that were made during development. I mean, there's no way to go back, obviously. And so resilience has to do both with genetics as well as development, and of course the stimuli that you have. We're now working on different kind of protocols, how to increase resilience, but it will require some more work about the specific patterns of stimulation that can actually help promote this. It's a big topic. Yeah.
Thank you, Ronny. As I mentioned before, we have a very short time, and we are in the end of our webinar. Now it's 11 o'clock in Brazil. Thank you, Shulamit. Thank you, Ronny, for joining our webinar. It was very interesting, amazing presentation, and I'm sure that we will see each other and in other events together soon. Okay? Stay well, take care, and thank you very much. Mário, Salomão, muito obrigado por tudo isso, por facilitar esse evento, esse webinar fantástico, todos que ajudaram, Sila Schumann, que organizou em nome da Câmara e das entidades também, muito obrigado. E isso é Israel, isso é a mensagem que Israel tem para o mundo, e é o que a gente tenta desenvolver aqui no Brasil, da forma que a gente pode, da forma que a gente consegue, mas com muita vontade, assim como o Mário, pelo Weizmann, Salomão pelo Technion, nós pela Câmara Brasil-Israel e tantas outras entidades que participam, colaboram e apoiam o Estado de Israel nas suas iniciativas, juntamente com o Brasil. Então, muito obrigado a todos e tenho a certeza que nos vemos em breve. Thank you very much. Bye.
Bye-bye.
Bye-bye.
Thank you.